Development is the key. This will be about growing a creature, not sculpting one. However, before anyone shouts genetic engineering and DNA, my first call is that we can't use human genetic material because that dodges the issue. The textual Moreau was determined to create humans without using human sources at all, sticking to what the non-human animals provided singly or in combination. The same applies here, although I can rephrase it in more rigorous terms. Remember, this isn't an engineering experiment to get a human from a non-human as cheaply and easily as possible, so it's not about genes and closest possible relatives. Instead, it's about the plasticity of development. So discovering whether genetic differences operate as a constraint is part of the point. The real beauty lies in the observation that animal morphogens are apparently closely conserved throughout evolutionary history. In other words, although you and the nearest fruit fly differ drastically in body materials and form, you and it used very similar physiological devices to organize your bodies. This conservation goes all the way down to the genetic level. If you restrict your attention to the mammals alone, then we are talking about vastly different outcomes from what amounts to minor differences in the use of nearly identical toolkits. This makes everything so much easier in some ways. It means that a creature's DNA is not a magic blueprint for all its features. Some traits, a very few, can each be traced directly to a single gene, such as the components of our immune system and our blood types. However, the majority are best thought of as participants in chains of biochemical effects operating in steps called cascades. Different cascades occur at different times and at different locations in the embryo, influencing the ongoing development. Fortunately, the remarkable diversity of mammals apparently represents few variations on a limited range of recipes. Given a few nudges for yay or nay and a few morphogens and conditions at the right places and times, and you could well have developed into a muskox. Using human development as a map and schedule for morphogens release and similar effects like apoptosis, tweaking a non-human mammal into a different mammalian species configuration may well be a matter of finding what morphogens to drip in or to inhibit and where and when. So the watchwords become location, 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 and conditions, 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 as well as knowing whether a particular outcome or detail gets socked into place early versus what is due to twiddling later. Even if you have the right morphogens at the right place at the right amount, you don't want to introduce it at the wrong time. The point is to mess with the cascade of events in the right way to induce the rest of the cascade operating normally to do what you wanted without further need to mess with it. Another procedural parameter is to consider whether you want to change the substance affecting the tissue or the tissue's response to the substance. If this is striking you as impossible, well, it pretty much is at present. But to focus on one element, bear in mind how much more we know about the genes compared to even 20 years ago. Both human and rat genes have been fully sequenced, and one of the big game changers of recent years is called genomics, the technology that permits not only mapping the entire range of genes of an organism, but also measuring their activity. Since the real meat of development lies not in single genes and single proteins, but complex interactions among them, the new technology makes it at least conceivable to measure such interactions as epistasis, heterosis, and other such jargon-named events directly, which is exactly what this project would rely upon for its initial procedural designs.